In our last video we have stopped at various parameters that are affecting the equilibrium of the protein dioxygen binding. Now let us take one more parameter. This is a different parameter from all these things. It is not about the equilibrium, but we are trying to compare the fetal hemoglobin with that of the mother hemoglobin. From this diagram we can see that fetal hemoglobin has a greater affinity towards dioxygen. And why it is having a greater affinity means, in hemoglobin, when it is not bound to dioxygen, the sixth coordination site is taken up by either water molecule, or it could also be taken up by 2,3-biphosphoglycerate. This 2,3-biphosphoglycerate is weakly bound in the case of fetal hemoglobin. So once it is weakly bound, that will create a driving force for the dioxygen to come, so that the fetal hemoglobin will have a greater affinity for dioxygen. So this is the reason why the oxygen can very easily travel from mother's blood towards the fetal blood. So to conclude, fetal hemoglobin has a higher affinity for dioxygen, whereas mother hemoglobin comparatively has a lower affinity for dioxygen. So let us conclude all of these oxygen saturation curves. Suppose if I choose this as the ideal curve, with this ideal curve above and below, if all these parameters are taken, pH increases, it will be above. If pH decreases, that curve will be below the ideal curve. When it comes to the temperature, above the ideal curve will be a low temperature curve, whereas below the ideal will be high temperature curve. Two parameters. Let me also take the third parameter. Above this ideal curve will be low CO2, below this ideal curve will be high CO2. So this is how we can conclude the oxygen saturation curves. Now previously we were discussing about the Hill's constant. Hill's constant N for hemoglobin, it is found to be around 2.3 to 3 depending on the organism. For humans it is about 2.8. So let us just see the percentage saturation for hemoglobin, as the Hill's constant is found to be 2.8 for human beings. If at all we calculate the percentage saturation, there are 4 subunits and Hill's constant is 2.8. So if I take its percentage, it can never reach 100%, it will always be lesser than 100%. Whereas for myoglobin it is a monomer, there is only a single unit and the Hill's constant is found to be 1. Therefore if at all we take its percentage, it will be 100%. Since myoglobin can reach 100% saturation, it can better act as a storage protein, whereas hemoglobin, it can never reach 100% saturation, so it can better act as a transport protein. Hence hemoglobin do not act as a storage protein. The same it is also given here. When hemoglobin is reacting to 4 dioxygen molecules, we can see the order of the dioxygen being 2.8, whereas for myoglobin it is 1. Now also, when hemoglobin, once it is getting oxygenated, as it is going to the tissue level or the cellular level, it reacts with carbon dioxide and water. For the 4 oxygen molecules that it is giving, every hemoglobin will take 2 protons, as these protons are attaching at various coordination sites. There will be some conformational changes. So as the protons have started to attach to the hemoglobin, that brings some conformational changes, and these conformational changes starts to release dioxygen. Carbonic acid is also formed. So much of the carbon dioxide is being removed in the form of carbonic acid. Also, whatever CO2 that is released, it can directly bind to hemoglobin. Nearly about 10% of carbon dioxide can directly bind to hemoglobin, and such a hemoglobin is called as Carbaminohemoglobin. It is also called as carb hemoglobin, carbohemoglobin, or even carbaminohemoglobin. So this is about the process of cooperativity and Hill's constant. So just let me conclude these points. Deoxyhemoglobin, the iron is present outside the plane of the porphyrin. But as it is taking up dioxygen, slowly the iron will enter into the plane of the porphyrin. So in the deoxy form, the sixth coordination site is either occupied by a solvent molecule, which is a water molecule, or it could bind to the 2,3-DPG or a 2,3-BPG. The binding could be weak or it could be moderately strong. As it is binding to dioxygen, the unbinding of 2,3-DPG will create a driving force for the binding of dioxygen. When dioxygen binds to iron, stability for the iron dioxygen bond will come from the hydrogen bonding from a distal histidine. Such a oxyhemoglobin will slowly travel from the lungs towards the tissue at the cellular level. At the cellular level, the partial pressure of dioxygen decreases. Carbon dioxide environment will be higher. Carbonic acid will be forming. Protons get attached to the various coordination sites of the hemoglobin. That brings some conformational changes in the hemoglobin. Once conformational changes appear in hemoglobin, it starts to lose dioxygen, whatever it is binding to, and slowly it will take up the carbonic acid. It binds to the carbonic acid, and that carbonic acid will again come to the lungs. That carbonic acid is back converted into carbon dioxide, which is expelled out. Also, nearly about 10% of carbon dioxide can directly attach to hemoglobin, which we call it as a carbo or carbaminohemoglobin. So this is how the process of respiration occurs, where the hemoglobin is acting as a oxygen carrying protein.